Hello, my dear grade 10 students. So at this point, we're going to talk about heredity, inheritance, and variation. So the last time you learned about this topic is when you were in grade 8. So in this session, we're going to dig deeper into the secret of heredity, secret of genetics. And to start with, so I understand that you are too familiar with this figure, this diagram. Okay, so it's been a year that we were affected by this coronavirus. And lately, a variant was discovered in UK, Africa, and other parts of the world. So why is that possible? Okay, very good. It's because of mutation. And why are we so concerned about this COVID variant? Okay, so how concerned should we be about the new variants of SARS-CoV-2 which cause COVID-19? Is it unusual for the viruses to change and mutate? Do vaccines protect against these variants, and what can you do to protect yourself? So speaking of vaccines, so these vaccines that were in the market is intended for the old variant. So since there is a new variant, there will be another discovery of vaccine. And of course, because this is somewhat infectious compared to the old variant, what are the means and ways on how you can protect yourself? No, so we are so threatened by the existence of this COVID variant. Okay, so for this lesson, we are going to explain how protein is made using information from DNA. Explain how mutations may cause changes in the structure and function of a protein. And in the end, we are going to present an output describing the effect of DNA mutation in organism and its impact to humanity. So I understand that you've heard a lot about the discovery of this DNA. So in history, you come across with the name Gregor Mendel and a lot more. And when Gregor Mendel experimented on pea plants, he was able to conclude that this DNA is the genetic material. So it's responsible for the transmission of traits, passing of traits from parents to offspring. So what exactly is DNA? Okay, so if you're going to look at the structure of DNA, the composition, so here, obviously you see that DNA is like a twisted ladder. Okay, so this is made up of nucleotides, and this nucleotide is made up of the backbone, which is the phosphate molecule, and the sugar molecule. And of course, it is composed of one of the 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. And this adenine will always be paired to thymine, and guanine will always be paired to cytosine. That is what is written in Chargaff's rule by Erwin Chargaff. And this DNA is important because this is an example of nucleic acids. So we know that DNA stands for deoxyribose nucleic acid. So what do you know of nucleic acid? Okay, so nucleic acid is a large molecule compound of carbon, hydrogen, oxygen, nitrogen, and phosphorus. So these are the elements that you can find in the nucleic acid. And there are actually two kinds of nucleic acid. What are those? Okay, so we have DNA and RNA. So I want you to look at the figure here and figure out the difference between RNA and DNA. Okay, so first you observe the strand. One is single, DNA is a double strand. Okay, and then you look at the nitrogenous base. So here you have cytosine, guanine, adenine, and thymine. But in the RNA, the thymine is replaced by uracil. So this summarize the difference between DNA and RNA. So for sugar, DNA is deoxyribose, while RNA is ribose. So again, DNA is a double strand and RNA is single strand. So for nitrogenous bases, so uracil replaced the thymine in DNA. Found in the nucleus, the DNA, and RNA is found in the cytoplasm. And you look at their function, DNA serves as the blueprint of biological guidelines, and RNA assists in carrying out DNA's blueprint guidelines. So DNA serves as the blueprint. So it's like how architect, of course, no, before building a house, must have this blueprint, because this is important, because this, of course, gives the instruction on how a house should be built. Okay, so why are nucleic acids essential in heredity? Okay, so because nucleic acids are organic compounds that function as storage of genetic information, which is transmitted from one generation to the next in all living organism, whether it is a plant or an animal. Okay? But of course, before this gene information is transmitted, there is a complex process that needs to be done for this to materialize. And with that, what do you think is the process involved in copying of the genetic information and transmitting that from one generation to the next? Okay, so it involves protein synthesis. Okay, so what do you mean by protein synthesis? So this is a series of processes involved in the production of protein. Okay, and because this is a series of processes, what do you think are the processes involved in order for a protein to be produced? Okay, so it involves three processes. We have replication, transcription, and translation. So when we say replication, so from the word replica, okay, so we understand what the word replica means. So this is how DNA copies itself. So this replication is a process of producing two identical replicas from one original DNA molecule. So we're going to start with DNA. Okay, so when this original DNA produce two identical copy, and the process is said to be semi-conservative, for each contain two copies that each contain one of the original strand and one new strand. So an exact copy of the original. So it is said that the process is semi-conservative. And you look at this diagram, The video is already provided in the LMS. So if we are to make the process short, if we want to simplify, here you will see that you'll find so many enzymes. So these enzymes help in the process. In order for the process to continue to work, so there are a lot of enzymes required. So here we have the original template of the DNA, and it will start with unzipping or unwinding in the area we called replication fork. And the unwinding and unzipping is performed by the helicase enzyme. Okay, so when this original template separates into two, it's like you have unzip a zipper. Okay, so here you see that second, the process is the complementary base pairing. So we understand that in Chargaff's rule, the adenine will be paired to thymine and cytosine will be paired to guanine. So that's the joining with the help of the DNA primase. And of course, the DNA polymerase will do the joining. Okay? And at the end of the process, a product here from the leading strand and from the lagging strand, we expect that this will produce two copies of similar to the original DNA. Okay, so the replication is summed up into first unwinding and unzipping with the help of enzyme helicase, complementary base pairing with the help of primase, and of course the last is joining with the help of DNA polymerase. Okay, so what is the importance of DNA replication to DNA's function of storing genetic information? So take note that here we are after with storing genetic information. That's the role of the nucleic acid. But what is exactly the significance of replication to storing genetic information? So this DNA replication is compared to a blueprint. So from the table, we see that DNA serves as the blueprint. And since it contains the instruction to construct other components of the cell. And this DNA replication is important in regulating the expression of genetic information. So we called it as gene expression. And it is preserved and pass the same information found in the DNA. So it serves as a blueprint and important, very important in gene expression. So what do we mean by gene expression? Okay, so this is the process by which information from a gene is used in the synthesis of a functional gene product. So information from a gene is used in the synthesis of functional gene product, and that functional gene product is primarily a protein. So how is that DNA is translated into protein to that functional gene product? Okay, so for gene expression to take place, so how do our cells accomplish that gene expression? And how do genes, which are found in the DNA, give rise to actual traits or characteristics? So this will include or will move on to the next process after replication. So it's from DNA to RNA. So but before we go to the next step, we understand that RNA is entirely different from DNA. Okay, so from the table you see the difference. So for the structure of RNA, it is again a single strand, and you see the base here is uracil instead of thymine. And there are three types of ribonucleic acid. So the first one is the messenger RNA or mRNA. So what's the function? It encodes amino acid sequence of a polypeptide. So we'll check the role of these types of RNA in protein synthesis. Then we also have transfer RNA or tRNA. This brings amino acids to ribosomes during translation. And we have also ribosomal RNA or rRNA. This makes up the ribosome, and this is the organelles that translate the mRNA. Okay, so let's check how these types of RNA works in protein synthesis. So after replication, take note, we learned that in replication, the original template of the DNA produces two identical copies, and this, of course, serves as the blueprint for the genetic information. Okay, so we'll be moving on to the next, which is transcription. So what do you know of transcription? Okay, so this is all about making working copies of the genes. How is that possible? Okay, making working copies of the genes. Take note, genes are in the DNA. So how transcription make a copy of this gene? So this is the first step of gene expression. So for gene expression, it's about how it will form a functional gene product, which is a protein. So here, this is a particular segment of DNA is copied into RNA, or we call it as messenger RNA, by the help of an enzyme, RNA polymerase. So a particular segment of DNA is copied. Take note that DNA can't move out of the nucleus. So the segment of DNA is copied, and that is the mRNA or the messenger RNA, that segment of DNA. And this is a process by which genetic information in DNA is transferred into an RNA molecule. Okay, so why it is transferred to an RNA? Because DNA can move out of the nucleus. So for that genetic information to be passed, so it's transferred into ribonucleic acid. So that's the mRNA or messenger RNA. So this is the steps involved in transcription. So here, transcription produces a single-stranded molecule of RNA. Take note, it's a single strand. One strand of the DNA will serve as the template. So if this is the DNA, one strand of it will serve as the template. So transcription starts when the DNA molecule, this is the DNA molecule, open up along a gene. Okay, so that gene is actually part of the DNA. The RNA nucleotides, which is the adenine, uracil, cytosine, and guanine, match up and join the open DNA. So this is the matching. Okay, and then the complete RNA strand, this one, is released and it will move into the cytoplasm. So this is the newly made RNA. A single strand that moves out moves to the cytoplasm because the DNA can move out of the cytoplasm. So DNA is transferred to RNA. So this is the messenger RNA. Okay? So it's about opening the DNA molecule, base pairing of the nucleotides, and then RNA strand is released into the cytoplasm. Okay, so that's for transcription. So from that transcription, we see how DNA is transcribed into RNA. So in our replication, we see that DNA makes a copy of itself. That's why it's DNA to DNA. Then in transcription, the DNA is transcribed into RNA or transferred into RNA or in messenger RNA. So in translation, it's the process of making the protein. So how this works. So this is the last stage in gene expression. And it is the process in which cellular ribosomes create protein. How? So let's see, because ribosomes are found in the cytoplasm or located in the cytoplasm. So this messenger RNA, which is the product of the transcription, of is decoded by a ribosome to produce a specific amino acid chain, or we call it as polypeptide. Okay, so for the steps in translation, ribosome binds with the mRNA. So this is the one that moves into the cytoplasm, product of transcription. And then this messenger RNA will be the codon. So we'll check what a codon is. This will match with the anticodon or the transfer RNA. And the amino acid carried by this transfer RNA add to the polypeptide chain. And ribosome, when hits the stop sequence, it releases the polypeptide and the messenger RNA. Okay, so speaking of codon, this is composed of a sequence of 3 nucleotides in messenger RNA. So that messenger RNA base pair with the anticodon in sequence of 3 nucleotides. Okay, not a single nucleotide, but in groups of 3 or sequence of 3. So that's the codon. So the anticodon is a sequence of 3 nucleotide bases that can base pair with a specific messenger RNA codon. Okay, so this is the transfer RNA. So since this is composed of 3, a sequence also of 3 nucleotide base pair with the messenger RNA codon. And for you to check that amino acid, so we are going to check how or what kind of amino acid is formed. So how a message from the RNA is translated into protein. So these are the amino acids, okay? And a chain of this will construct a protein. So for the messenger RNA transcribe code, or we call it as codon. So in groups of 3 or sequence of 3 nucleotides. So since uracil will match with adenine and guanine with cytosine, so this will be the anticodon that will represent valine. That's the amino acid. So for this group, for this sequence, this will be the anticodon, and that will represent as the glycine. And for this 3 sequence, for the code, this will be the anticodon, and that will stands for methionine. Okay, so adding of 3, and then this will add to the chain. Okay, and then when it hits stop, it will produce the protein. So for you to check the amino acid, you are going to make use of the genetic code. So what is this genetic code? This is a universal language that translates the gene and the amino acid, which matches the 3-letter combination of the 4 nitrogenous bases to its equivalent amino acid. And this is the table for the genetic code. So this consists of 64 code wherein it's made up of 3 letters. So the first base is found on the right, the second base is found on top, and the third base is found on the right. And with letter combination, say first letter here, how is this form? U, second is U, and the last is U. Okay, so this will be the amino acid. Okay, so if we have C and then U and then U, so we have C, U, and U. So that's leucine. So there are lots of amino acid. We have valine, alanine, proline, serine, arginine, and a lot more. Okay, so you check the exact name of these amino acids. Okay, so with that, the protein synthesis is actually simplified into how you make bread. So we started with DNA, it's transcribed to RNA, and it's translated into protein. How? If the cookbook represents the DNA, it contains the instruction, the genetic information. So since it can move out of the nucleus, so the copy of this, which is taken from the cookbook, will represent as the RNA. So this is the exact copy of what is in the cookbook. Okay? So that's transcription. So in making the bread, so that cookbook actually produces the bread. So upon following the instruction of this, what is written in this recipe, you will create the bread, and that will represent as the protein. So that's in the process of translation. Okay, so the central dogma of molecular genetics is simplified in this one. So in replication, so since this is replication, an exact copy of the DNA, that's why DNA produces DNA with the help of DNA polymerase. Then in transcription, the DNA is transcribed into RNA with the help of RNA polymerase. And in the translation, the RNA is translated into protein. So it's because of the help of the ribosome. Okay? So for you to easily understand protein synthesis, you just remember this part. Okay? So next is all about, again, going back with the problem on COVID variant. So you know that this is brought about by mutation. Okay? So when we say mutation, there's a change in the molecular structure of a gene or the physical structure of the chromosome. And this is brought about by mutagens. So mutagens are agent that causes mutation. And mutation occur because the DNA fails to copy accurately. So that's the importance of this semi-conservative replication, wherein the information, the instruction is actually copied. And when copying is not accurate, then it will result to mutation. So what are the causes of mutation? Okay, first is smoking can raise risk of genetic mutation. And then we have also effect of old age on offspring. Chemotherapy, so it's all about the effect of radiation. And of course, other factors, external influences. So what are the different kinds of mutation? We have gene mutation. There are two under gene mutation. We have point mutation and frameshift mutation. For chromosomal, we have lots duplication, translocation, inversion, deletion, and insertion. So let's take a look at this kind of mutation. So the first is point mutation. So here, single nucleotide base is changed, inserted or deleted from a sequence. So this is the original. So here, the base is actually changed. Okay, so with that, it's point mutation. And then for frameshift, this is also called framing error or reading frameshift. Here, a number of nucleotides in a DNA sequence that is not divisible by 3. Okay, so we understand that the sequence for the codon or messenger RNA are into sequence of 3 nucleotides. So if it's not divisible by 3, so it will result to frameshift mutation. Then we also have for chromosomal, these are the examples. So for insertion, obviously, literally from the word insert. So another in a genetic material added another chromosome. So here chromosome 4 is inserted to chromosome 20. Okay, we also have translocation when part of the chromosome breaks off and is combined to another chromosome. Okay, chromosome 20, look at what had happened after translocation. It combines to another chromosome. Then for deletion, from the word delete, a part of the chromosome is lost in the process. Okay, so this is what happened after deletion. And we also have duplication. Here, a part of the there is an extra copies of a part of the chromosome. And for inversion, when the direction of a part of chromosome is reversed. So it's the orientation of the chromosome. And with all this, of course, again, this mutation is brought about by smoking, exposure to radiation in particular. Kaya when you heard about the Chernobyl incidents where this is still a problem nowadays because of the radiation greatly affect specifically the health of those who were exposed or who have exposure to radiation. And there are a lot of diseases, of course, disorders that are possible to experience once there is problem with mutation. So we have recessive disorder, sex-linked disorders, and human genetic syndromes. And these are the examples among the many examples of disorders related to genetic mutation. And we know that when we are dealing with an individual having this kind of disorders, we really are too careful in dealing with them because we understand that they are not the same with us. They are very different and we need to accept their condition. Okay, so that ends our discussion. So I hope you get something from this lecture video. So see you next session. Thank you.